Imagine, if you will, that you are just, eagerly tracking a highly anticipated, incredibly precious package in the mail.
Oh, we've all been there. Constantly refreshing the tracking page.
Exactly. You're checking the app every single day. But instead of an Amazon delivery van pulling up to your driveway, uh, this particular package falls directly from the sky.
Literally drops from orbit.
Right. It streaks through the atmosphere, glowing white hot, and it crashes into the remote Australian outback. The date is December 2020. And the precious cargo inside this, multi-million-dollar delivery, it's a literal pinch of dirt.
Just a tiny speck of dust.
Yeah. But that dirt, which was retrieved from an asteroid millions of miles away, well, it contains a chemical secret that connects the cold vacuum of space directly to the DNA inside your own body.
It is genuinely mind-blowing.
It really is. So welcome to the Deep Dive Lab. Today, we are exploring a breathtaking discovery that traces the very alphabet of your genetic code back to the dark, freezing dust of our early solar system.
And, to understand how we found this profound chemical connection, we're diving into a landmark research paper. It was published in March 2026 in the journal Nature Astronomy.
Yeah, alongside that really excellent supporting analysis from Chemical and Engineering News.
Right, exactly. And together, these pieces document how researchers found the ultimate building blocks of life in a microscopic speck of space dust, and what that reveals about the chemical pantry of our early solar system.
Which is just, it's wild. And to really appreciate the magnitude of what was found inside that little capsule in the Australian outback, we have to start with a massive, decades-long headache.
Oh, a massive headache.
Right. Astrobiologists have been wrestling with this for years. It revolves around something called the RNA world hypothesis because, well, when we talk about the origin of life, we have a kind of chicken and egg problem.
A classic paradox, yeah.
Exactly. Today, you have DNA to store your genetic blueprints, and you have proteins to actually execute the chemical work in your cells.
Right, the laborers.
Yeah. But DNA can't build things, and proteins can't store information, so how do you start?
And that is the core of the puzzle. Because billions of years ago, before that highly specialized two-part system evolved, early life needed a molecule that could do both.
Like a multi-tool.
Exactly, a biological Swiss Army knife.
Right.
And scientists strongly believe that early life relied primarily on RNA.
Right.
RNA is incredible because it is built from these fundamental chemical letters, nucleobases. Specifically, adenine, guanine, cytosine, thymine, and uracil.
The famous A, G, C, T, and U.
You got it.
Yeah.
And RNA can both carry genetic information like a hard drive, and it can fold up to spark chemical reactions like a biological machine.
So it does all the heavy lifting early on.
Exactly.
So if RNA is the bridge to the very first spark of life, finding out where those 5 specific nucleobases originally came from is, it's basically the holy grail of astrobiology.
Oh, absolutely. We need to know where the factory was that manufactured those initial ingredients.
Right, but this is where the historical headache comes in, right? Because for a long time, researchers have been analyzing meteorites that crashed down to Earth looking for those ingredients.
Yeah, like the really famous Murchison meteorite.
The one that fell in Australia back in 1969.
Right. And over the years, scientists have actually found some of those nucleobases inside it.
Wait, okay, hold on. I have to push back here. If we've had the Murchison meteorite sitting in a lab since 1969, and we already found nucleobases on it, why are we spending hundreds of millions of dollars to drop a new package in the desert?
It's a fair question.
Right. Isn't a space rock just a space rock? It seems like we already had the answer decades ago.
Well, the problem really comes down to the chain of custody. Think about the environment we live in, right?
Right.
Earth is absolutely teeming with life.
Just covered in it.
Exactly. So from the moment a meteorite hits the atmosphere, it is bathing in Earth's biosphere. It strikes the soil, which is packed with microbes and fungi. And then it sits in a museum where humidity carries organic aerosols. It might even pick up a tiny trace of human DNA from a researcher's breath.
So it's a massive contamination risk. It's like dropping a sandwich on the kitchen floor and then trying to figure out if the dog hair was already on it.
That is exactly it. If you crush up a meteorite that's been on Earth for 50 years and you find an adenine molecule, you have no way to definitively prove that molecule formed in the vacuum of space.
Because it could easily just be the chemical remains of an Earth microbe that, hitched a ride after the rock landed.
Precisely. And the scientific community recognized that as a fundamental roadblock. To definitively prove that the universe is manufacturing these molecules abiotically.
Meaning completely without biology.
Right, completely without biology, just out in the cold vacuum of space. They couldn't rely on rocks that fell to Earth on their own. They needed a perfectly pristine sample,
Untouched.
one that had never touched Earth's environment, never breathed Earth's air. They needed to go out and collect the dust themselves.
Which brings us to the Japan Aerospace Exploration Agency, or JAXA, and their primary Hayabusa 2 mission.
A truly incredible mission.
It really was. They flew a spacecraft to a carbon-rich asteroid named Ryugu, grabbed a sample directly from the source, sealed it up tightly in the vacuum of space, and dropped it back to Earth in 2020.
They bypassed the floor entirely and went straight to the cosmic pantry.
I love that. But securing that pristine, untouched sample, that just shifted the challenge entirely, didn't it?
Oh, definitely. The space exploration part was a triumph, but now the burden fell to the laboratory.
Right, because possessing the dust is only step one.
Exactly. The new problem was how to find completely invisible organic molecules in a sample that weighs less than a raindrop.
So let's introduce the astrochemist who took on this staggering puzzle. Dr. Toshiki Koga from the Japan Agency for Marine Earth Science and Technology.
Yes, Dr. Koga.
In 2023, JXA opened up a call for proposals to analyze the Ryugu dust. Dr. Koga and his team applied to conduct a comprehensive search for nucleobases.
And they were granted a sample.
Right. But we really need to emphasize the scale here. They were given about 20 milligrams total.
It's almost nothing.
It's roughly the weight of a single grain of rice, chopped up. And that tiny amount was split between a sample taken from the asteroid's surface and another taken from deep in its subsurface.
And working with a sample that small, it requires an almost unbelievable level of precision.
I can't even imagine.
Yeah, Hannah L. McLean, who is an astrochemist at NASA and reviewed the study, she was deeply impressed by the methodology Koga's team developed.
What did she say about it?
Well, the goal, as McLean put it, was to eke out every single particle and every piece of data from that minuscule amount of material. You just can't afford to waste a single molecule.
I'm imagining you can't just crush a 20 milligram sample and put it under a standard microscope.
No, definitely not.
If you are looking for these delicate organic molecules, do you just, steep it like a microscopic cup of tea to draw them out?
That is actually very close to the first step of their sequential extraction process.
Wait, really? A space tea?
Basically, yeah. They started by bathing the dust in hot water. Water is a fantastic, gentle solvent.
Okay, that makes sense.
It pulls out the loose, water-soluble organic molecules that are just clinging to the surface of the dust grains, and it does so without destroying their chemical structure.
Which is key.
Right. But hot water only gets you so far.
Right, because asteroids aren't just loose powder. They are solid mineral matrices. Water isn't going to penetrate the rock itself. So what was step 2?
Step 2 involves taking what is left over from the water bath and treating it with strong hydrochloric acid.
Oh, wow. Acid.
Yeah, the acid is crucial because it actually eats away and dissolves the tougher mineral matrix of the asteroid dust.
Okay, so it breaks down the rock.
Exactly. By breaking down the rock itself, they released any molecules that had been tightly trapped inside the material during the asteroid's formation billions of years ago.
And once they extracted those fluids, they ran them through high-resolution mass spectrometry, which is basically a highly advanced scale that weighs individual molecules down to the extreme decimal point of their atomic mass.
Yeah, it allows the team to identify exactly what was floating in that liquid.
So what did they find?
Well, the results they got back from that mass spectrometer were nothing short of a jackpot.
A literal scientific jackpot.
Yes. They didn't just find 1 or 2 biological building blocks. They found all 5 biologically relevant canonical nucleobases across the samples.
There's all 5.
All 5. Adenine, guanine, cytosine, thymine, and uracil. The complete chemical alphabet required for life, sitting right there in this tiny pinch of asteroid.
It is staggering when you really picture it.
It is.
And the inventory didn't stop there, did it? They also found vitamin B3, which is nicotinic acid, along with various amino acids and surprisingly high levels of urea.
Yeah, really rich chemical soup.
But what I find really compelling is the spatial distribution of those molecules.
Right, the surface versus subsurface.
Yeah. The subsurface sample, the material that had been shielded deep inside the asteroid, protected from the harsh, degrading radiation of deep space. It actually contained 3 times the total nucleobase concentration compared to the surface sample.
The shielded environment of the subsurface clearly preserved a much richer chemical inventory.
That makes total sense. But even with that amazing data, we still have to address the elephant in the
The
room.
contamination question.
Exactly. Because the initial problem we discussed was Earth contamination. And even though JAXA brought this capsule back sealed from space, skeptics could still argue that, maybe Earth-based molecules somehow slipped in during the lab extraction process.
Sure, you always have to rule that out.
Let's tackle that. How do we know without a shadow of a doubt that these molecules aren't just the result of a rogue microbe in Dr. Koga's lab breathing on the dish?
The definitive proof lies in what Dr. Koga's team discovered alongside the standard nucleobases. They found something called structural isomers.
Structural isomers.
Yes. A prominent example in their data is a molecule known as 6-methyluracil.
Okay, let's pause and visualize what a structural isomer actually is for everyone listening.
Good idea.
Imagine you are building a structure with Lego bricks. The standard nucleobases, the ones that life on Earth uses, they are like perfectly stamped standard Lego pieces.
Right.
Every biological cell on Earth acts like a highly specialized factory, using specialized machines called enzymes to manufacture those exact shapes so they click together perfectly to form an RNA or DNA strand.
That is the perfect way to frame it. Earth biology is incredibly picky. Enzymes are designed to build only the correct shapes because anything else would break the genetic chain.
So if Earth is pumping out standard bricks, 6-methyluracil is like finding a Lego brick made of the exact same plastic, weighing the exact same amount, but the little bumps on top are shifted into entirely the wrong places.
Yes, it won't click into a biological DNA strand.
Right. Since Earth biology only mass produces the standard bricks, finding this weird misaligned isomer in the Ryugu sample proves it wasn't made in an Earth-based biological factory.
The logic is undeniable. Space chemistry isn't driven by neat, highly specific enzymes. It is driven by random blind forces.
Like radiation just mashing atoms together.
Exactly. And that random process creates every possible shape, including those misaligned isomers.
So it's a messy process.
Very messy. Finding 6-methyluracil is the ultimate chemical fingerprint. It confirms that these molecules formed completely abiotically through random chemistry in the vacuum of space.
Okay, so we have undeniably proven the origin. But here's where the research takes a really fascinating turn.
Oh, it gets so good here.
Because finding the molecules was a monumental triumph. But it was the exact ratio of these molecules that revealed a completely unexpected twist about how our solar system was baking these ingredients.
Yeah, let's look at how those 5 nucleobases are categorized. They are divided into 2 structural families. You have the purines, which include adenine and guanine, and you have the pyrimidines, which include cytosine, thymine, and uracil.
Purines and pyrimidines, got it.
Dr. Koga's team calculated the ratio between these 2 families in the Ryugu samples, and the balance was striking. Ryugu has a nearly equal ratio of purines to pyrimidines, sitting right at about 1.1 to 1.2.
Let me make sure I'm picturing this correctly. A 1.1 to 1.2 ratio means they are practically balanced 1-to-1.
Exactly 1 to 1, yeah.
But when we look at the broader catalog of space rocks, is that balance normal?
It's actually highly unusual.
Really?
Yeah. When researchers analyze the Murchison meteorite, they find it is heavily enriched in purines. The scale tips drastically to one side.
Okay.
But if you look at samples from the asteroid Bennu or the Argyll meteorite, the scale tips drastically to the other side. They are heavily enriched in pyrimidines.
So Ryugu sits uniquely in the middle.
It does.
But wait, if all of these ancient rocks are floating in the same solar system and forming around the exact same time, why are their chemical pantries stocked so differently?
That is the million-dollar question.
Right. What is driving one rock to mass-produce purines while another mass-produces pyrimidines?
This is where Dr. Koga's team uncovered a stunning correlation across all these different extraterrestrial samples. The ratio of purines to pyrimidines perfectly negatively correlates with the concentration of ammonia.
Ammonia, like the household cleaner.
Yeah, exactly.
We usually think of that as a biosignature we look for on exoplanets or just a planetary gas. But you're saying ammonia was acting as a primary chemical reagent inside these ancient rocks.
It was acting as the master switch. Billions of years ago, as these parent asteroids were forming, they accreted ice from the cold outer edges of the solar system.
Okay, picking up space ice.
Right. Some swept up a lot of ammonia ice, others grabbed very little. And the data shows that if an asteroid had a high concentration of ammonia, that environment strongly favored the production of pyrimidines.
And if it had low ammonia?
If the ammonia levels were low, the chemical pathway shifted entirely. The environment favored a process called hydrogen cyanide polymerization.
Hydrogen cyanide polymerization. Let's break down that polymerization process because hydrogen cyanide sounds incredibly toxic to us today.
Highly toxic, yes.
But it was apparently vital back then. How does a poison turn into a building block of life?
Well, inside these dark, freezing rocks, you had radioactive elements decaying.
Oh, okay.
And that decay released high-energy radiation, like gamma rays, providing a steady source of energy.
Like a cosmic oven.
Yes. In a low ammonia environment, that gamma radiation hits molecules of hydrogen cyanide, causing them to link together like a daisy chain.
Right.
As this chain of carbon and nitrogen atoms grows, the energy forces it to fold in on itself, eventually forming the complex, double-ring structures that we recognize as purines.
So it literally is a cosmic bakery.
It really is.
The amount of ammonia in the initial dough acts as a chemical switch, dictating whether the radiation bakes single-ring pyrimidines or double-ring purines. That is chemistry operating on a staggering solar system-wide scale.
It completely reframes how we view the early solar system. We have essentially reverse-engineered the chemical factories of the cosmos, figuring out not just what they made, but the exact recipes and conditions they used to make them.
Wow. Okay, so with the mystery of how they formed solved, we really need to zoom out and connect this to the bigger picture.
Always important to zoom out.
Why does it matter to you listening right now that an asteroid millions of miles away has a balanced ratio of DNA building blocks?
It matters deeply because of a process called exogenous delivery.
Exogenous delivery, what is that?
Well, if we rewind billions of years, the early Earth was an incredibly volatile, turbulent place. But it wasn't a closed system. It was subjected to a period of heavy bombardment.
Just getting absolutely pelted.
Right. Millions of carbonaceous asteroids and meteorites rocks with the exact same chemical makeup as Ryugu were raining down on the planet.
So they were acting as cosmic delivery vehicles.
They were stocking early Earth with a vast, incredibly rich, prebiotic chemical inventory. The complex molecules necessary to jumpstart life weren't just struggling to form in Earth's harsh early oceans. They were being delivered, pre-assembled by the ton from space.
That is amazing. But as exciting as that is, we probably need to manage our expectations just a bit.
Sure.
Finding the letters of DNA on an asteroid doesn't mean we found life itself in space.
No, that is a critical boundary to draw. The research highlights a brilliant perspective from Cesar Menor-Salvin, an astrobiologist at the University of Alcalá.
Oh, I loved his quote on this.
It's so good. He notes that finding nucleobases doesn't answer the whole origin of life question. Just as finding sand, clay, and rocks in a field doesn't explain the origin and evolution of architecture.
That is a phenomenal way to ground the discovery.
Isn't it?
Yeah. Finding these nucleobases is the difference between opening a pantry and finding flour, sugar, and eggs versus actually finding a beautifully baked, decorated cake. The asteroid impacts gave early Earth a fully stocked pantry. But our planet still had to do the complex, messy work of baking the cake. Those raw ingredients still had to organize, replicate, and evolve into the first living cells.
But realizing that the pantry was stocked from space fundamentally changes our understanding of human existence.
It really does.
For generations, humanity wondered if the building blocks of our genetic code were some freak, isolated accident. We wondered if it was a 1 in a trillion chemical fluke that only occurred in a very specific primordial puddle here on Earth.
And this Ryugu sample proves that just isn't the case.
No, it proves these molecules are widespread throughout the entire solar system.
The chemistry of life isn't an earthbound anomaly. It is a fundamental, active feature of the universe's natural processes.
It connects us to the cosmos in the most literal way imaginable. We are made of star stuff, as the saying goes, but we are also directly made of asteroid chemistry.
So let's take a moment to reflect on the immense journey we've mapped out today. We started with a tiny 20 milligram speck of asteroid dust falling into the Australian outback.
Barely enough to see.
Right. And from that single grain, using meticulous hot water and acid extractions, scientists pulled out all 5 canonical DNA and RNA nucleobases.
And they used the misaligned shapes of chemical isomers, like our weird Lego bricks, to prove beyond a doubt that these molecules were forged in the vacuum of space.
And by analyzing the ratios of those molecules, they discovered that cosmic ammonia levels act as a master switch, dictating the exact chemical recipes driven by gamma radiation inside these ancient rocks.
It gives you a completely different perspective on the night sky.
Doesn't it? Every time you look up at the stars, you aren't just looking at distant dead rocks and burning gas. You are looking at active, dynamic chemical factories.
Factories that, billions of years ago, forged the exact same chemical alphabet that currently makes up your own DNA.
It makes the vastness of space feel intimately connected to our own biology.
It really does.
And that leaves us with a final provocative thought for you to mull over. We now know that carbonaceous asteroids in our solar system are universally manufacturing these exact seeds of life.
Yeah?
And we know that these space rocks are constantly bombarding planets, not just here, but in solar systems across our entire galaxy.
Everywhere.
Right. So if the fundamental ingredients for life are this common and are automatically generated by the cold vacuum of space, just how crowded might the universe's pantry really be?
It is a profound question, and one that makes the universe feel a lot less lonely.
Thank you for joining us for this session in the Deep Dive Lab. If you want to keep exploring the incredible mysteries of our universe with us, be sure to follow and subscribe for more fascinating science stories unfolding in real time. Keep looking up and keep asking questions.